This is audio recording for visually impaired students. Energy production in a cell. Large food molecules contain a lot of potential energy in the form of chemical bonds, but it requires a lot of work to liberate the energy. Cell needs a quick, easy way to get energy for anabolism. This is done with ATP. ATP is an unstable molecule, the bonds of which are easy to break, making it a useful source of energy for cells. ATP gives ADP plus P plus free energy from food. Food energy plus ADP plus P gives ATP. Catabolic reactions generate energy to make ATP, and the ATP energy is used to drive anabolic reactions such as metabolic turnover, replacement of cell parts, growth and cell division, and special functions such as secretion, absorption, contraction, or signaling. Metabolism, the sum of all chemical reactions in the body, catabolism plus anabolism. All energy production begins in the cytosol of the cell. Large molecules are catabolized into smaller molecules, but very little energy is produced. Protein gives amino acid, triglyceride gives fatty acid and glycerol, carbohydrate gives short carbon chain. These similar molecules are then absorbed and processed in reactions inside the mitochondria. 40% of the energy is captured to produce ATP from ADP, and the remaining 60% escapes as heat used to maintain body temperature. Oxidative reductive reactions. Oxidation is removal of electrons, reduction addition of electrons. These reactions are always coupled. One molecule must be oxidized while another is reduced. The reduced molecule gains energy while the oxidized molecules loses energy. Cells more commonly perform dehydrogenation reactions where hydrogen is exchanged instead of a free electron, but this is still a redox reaction. Catabolism of large molecules results in reduced carrier compounds which are then oxidized to generate ATP. Generation of ATP involves the addition of a phosphate to ADP and can be accomplished one of two ways. 1. Substrate level phosphorylation. A high energy phosphate is transferred directly from a substrate to ADP, thus forming ATP. Oxidative phosphorylation. Electrons are transferred from an organic compound to a cofactor carrier molecule. The electrons are passed through other carriers to a final acceptor, and the passing of the electrons releases energy that is harvested to add a phosphate to ADP in a process called chemiosmosis. Carbohydrate catabolism. Carbohydrates are the primary source of cellular energy for most organisms. Glucose is the most commonly used carbohydrate and will always be used first. Fatty acid will be used second when glucose is in short supply, and then more rarely amino acids can be utilized. Glucose can be catabolized for ATP production in two ways. Cellular respiration requires oxygen to serve as the final electron acceptor in a series of redox reactions that generate ATP by oxidative phosphorylation. This is most efficient method of ATP production and involves reaction performed inside the mitochondria. 2. Fermentation requires an organic molecule to serve as the final electron acceptor and can be done in the absence of oxygen. ATP is synthesized using substrate level phosphorylation, which is less efficient in humans. This results in the production of lactic acid. Fermentation reactions are carried out in the cytoplasm. Aerobic respiration of glucose. C6H12O6 plus 6O2 gives 6CO2 plus 6H2O. 3 stages of aerobic respiration. Glycolysis. Oxidation of glucose to pyruvic acid with some ATP and NADH produced. Citric acid cycle. Oxidation of acetyl to carbon dioxide with some ATP, NADH and FADH2 produced. 3. Electron transport chain. NADH and FADH2 are oxidized, providing electrons for redox reactions, ultimately reduce oxygen to generate ATP. The majority of the ATP is produced at this step. NAD nicotinamide adenine dinucleotide and FAD flavin adenine dinucleotide are coenzymes that functions to transport electrons to the form of hydrogen. NAD positive carriers 2 electrons but only 1 proton. FAD carrier 2 complete hydrogen atoms, aka the Embden-Merov pathway. Glycolysis is an anaerobic process that occurs in cytoplasm. It consists 10-step metabolic pathway that catabolizes and oxidizes one glucose molecule into two pyruvic acid molecules and generates two molecules of ATP by substrate level phosphorylation. Many cells can survive on glycolysis alone, called fermentation, but it is not very efficient, and fermentation generates lactic acid as a waste product which will need to be removed and processed to prevent drastic alteration in pH and loss of homeostasis. Two stages. Preparatory stage. Two ATP molecules are used to phosphorylate one 6-carbon glucose and catabolize it into 3-carbon molecule. To energy conservation stage. The two 3-carbon molecules are oxidized to generate two 3-carbon pyruvic acid molecules. At the same time, two NAD positive molecules are reduced to two NADH molecules and 4 ATP molecules are produced by substrate level phosphorylation. Summary of glycolysis. 1 glucose plus 2 NAD positive plus 2 ADP plus 2P gives 2 pyruvic acid plus 2 NADH plus 2H positive plus 2 ATP. Decarboxylation. Preparation for the citric acid cycle. This is the first step in the aerobic process of glucose metabolism. This and all subsequent steps, citric acid cycle and electron transport, will occur only when oxygen is available and will take place inside the mitochondria. The 3-carbon pyruvic acid is decarboxylated into carbon dioxide and a 2-carbon acetyl. The acetyl is attached to coenzyme A, which serves only as a carrier, and one NAD positive is reduced to NADH. This will happen twice. One glucose generates two pyruvic acid molecule. Decarboxylation reactions occur in the matrix of mitochondria. Summary of decarboxylation. 2 pyruvic acid plus 2 NAD positive plus 2 coenzyme A gives 2 acetyl coenzyme A plus 2 CO2 plus 2 NADH. Citric acid cycle, aka Krebs cycle or tricarboxylic acid cycle. This part of the aerobic metabolism of glucose involves 8 enzymatic reactions occurring in mitochondrial matrix that function to reduce the coenzymes NAD positive and FAD. The 2-carbon acetyl is attached to a 4-carbon oxaloacetic acid, creating in a 6-carbon citric acid. Oxidation and decarboxylation reactions occur which catabolize the 6-carbon citric acid back into a 4-carbon oxaloacetic acid and 2 carbon dioxide molecules. At the same time, 3 NAD positive and 1 FAD are reduced into 3 NADH and 1 FADH2 respectively, and 1 ATP is produced by substrate level phosphorylation. Summary of citric acid cycle: 2 acetyl CoA plus 6 NAD positive plus 2 FAD plus 2 ADP plus 2P plus 4H2O gives 2 CoA plus 4 CO2 plus 6 NADH plus 4H positive plus 2 FADH2 plus 2 ATP. This is the truly aerobic part of the aerobic metabolism of glucose, as this is where the oxygen is utilized. Oxidative phosphorylation occurs on a membrane, the mitochondrial cristae, to generate most of the ATP produced from glucose. Coenzymes from the previous reactions pass electrons to a series of electron carrier molecules which carry out redox reactions, resulting in chemiosmotic generation of ATP. There are 3 classes of carrier molecules: FMN, flavin mononucleotide, protein plus flavin coenzyme; 2. Coenzyme Q, non-protein; 3. Cytochromes, protein plus an iron group. Events of the electron transport chain: NAD positive and FAD collect energy in the form of hydrogens from organic molecules during glycolysis, decarboxylation, and the citric acid cycle, becoming the reduced forms NADH and FADH2. 2. NADH and FADH2 are oxidized and pass hydrogens to the electron transport chain consisting of flavoproteins, cytochromes, and coenzyme Q. As electrons are passed along the chain, protons are pushed out through the membrane. This sets up concentration gradient with protons on the outside and electrons negative charge on the inside. 3. At the end of the chain, the electrons are accepted by oxygen, creating an anion inside, which has a strong affinity for the cations H positive outside. 4. Chemiosmosis generates ATP. H positive from the outside move towards O negative on the inside through special membrane channels that are coupled to ATP synthase, and the high energy diffusion of H positive drives the reaction ADP plus P gives ATP. The energy from one NADH from glycolysis can generate 2 ATP. The energy from one NADH from decarboxylation in the citric acid cycle can generate 3 ATP, and that from one FADH2 can generate 2 ATP for a total of 32 ATP. 5. H positive combines with O negative inside the mitochondria creating water. Summary of electron transport chain: 2 NADH from glycolysis plus 2 NADH from decarboxylation plus 6 NADH from citric acid cycle plus 2 FADH from citric acid cycle plus 6 CO2 plus 32 ADP plus 32 P gives 12 H2O plus 32 ATP plus 10 NAD positive plus 2 FAD. Final summary for aerobic respiration: C6H12O6 plus 6O2 plus 36 ADP plus 36 P gives 6 CO2 plus 6 H2O plus 36 ATP. In most eukaryotic cells, 36 ATP are produced from 1 glucose molecules, 2 from glycolysis and 2 from citric acid cycle by substrate level phosphorylation, and 32 from electron transport by oxidative phosphorylation. Glycolysis occurs in the cytoplasm, the citric acid cycle occurs in the matrix of mitochondria, and electron transport occurs on rest of mitochondria. Some energy is lost as electrons are carried from cytoplasm and across the mitochondrial membrane, and thus the NADH from glycolysis can generate only 4 total ATP instead of the 3 each observed for NADH created inside the mitochondria. Cellular respiration review: glycolysis. Anaerobic occur in cytoplasm. 1 glucose oxidized into 2 pyruvic acids. 2 ATP produced by substrate level phosphorylation. 2 NADH produced by reduction of NAD via oxidation of glucose. If no O2 available, pyruvic acid reduced to lactic acid fermentation. Erythrocytes gives glycolysis only. Skeletal muscle fermentation when no O2. Neurons and cardiac muscles cannot ferment, need O2. 2. Decarboxylation Krebs cycle, acid cycle. Occur in matrix of mitochondria. Decarboxylation: 2 pyruvic acid decarboxylated and oxidized into 2-acetyl coenzyme A plus 2 CO2 plus with 2 NADH. Citric acid cycle: 2-acetyl combined with 2 oxaloacetic acids creating 2 citric acid. Citric acid decarboxylated and oxidized 4 CO2, 6 NADH, 2 FADH2. 2 ATP generated by substrate level phosphorylation. Electron transport: aerobic occurs on crest of mitochondria. NADH and FADH2 reduced during glycolysis and citric acid cycle are oxidized. 32 ATP generated by chemiosmosis, oxidative phosphorylation. 12 H2O produced as waste from oxidation of oxygen. Electrons are passed to cytochromes, finally accepted by oxygen. With oxygen, 1 glucose will produce 36 ATP in most human tissue cells. Without oxygen, 1 glucose will produce 2 ATP in human tissue cells that are capable of fermentation, not neurons or cardiac muscles. Lipolysis, lipid catabolism. The process of lipid catabolism hydrolyzes triglyceride and the storage form of fat into glycerol and 3 fatty acids. The glycerol is converted into pyruvic acid in the cytoplasm and catabolized through the citric acid cycle in the mitochondria. The fatty acids are catabolized by the beta oxidation in the mitochondria to be entered into the citric acid cycle as 2 carbon fragments. For each 2 carbon fragments of fatty acid produced by beta oxidation, the cell can generate 17 molecules of ATP. This is 1.5 times the energy production when compared carbon to carbon as with glucose. Although lipolysis generates more energy, it requires more oxygen and occurs much more slowly than equal carbohydrate metabolism. Protein and amino acid catabolism. For proteins to be used for energy production, they must first be broken down into individual amino acids. Then for the amino acids to be used in citric acid cycle, they must have the amino group removed in a process called deamination, which requires vitamin B6. The amino group is removed in conjugation with a hydrogen creating ammonia, which is very toxic. The liver must then convert the ammonia to urea, which is relatively harmless for excretion by the kidney. The remaining amino acid carbon chains are then used at various stages in the citric acid cycle to generate ATP. The amount of ATP produced varies with the type of amino acids depending on which step the citric acid cycle it was entered. Catabolism of amino acid is not a practical source of quick energy and is typically only used in starvation situations. Proteins are harder to break apart than carbohydrates or lipids. Their catabolism generates toxic waste products, and they are the structural and functional parts of every cell, and thus tend to only be used when no other energy source is available. Instead, amino acids are usually simply recycled by hydrolysis of peptide bonds in one protein to be reassembled by dehydration synthesis into the next. Nucleic acid catabolism. DNA is never catabolized for energy. RNA can be broken down into simple sugars and nitrogenous bases. The sugars are metabolized in glycolysis, but only the pyrimidine bases, uracil cytosine, can be processed into the citric acid cycle. The purines, adenine, and guanine are deaminated and excreted as uric acid, making RNA metabolism very inefficient. Typically, nucleotides are simply recycled into new nucleic acid molecule and are not used for energy production. Thanks for watching.